Good morning, everyone. It gives me immense pleasure to introduce today's speaker, Dr. B. M. Gurupadaya. Sir is working presently as a professor and head Department of Pharmaceutical Chemistry at Vgst College of Pharmacy, Mysuru. Sir has completed his UG in 1995 from SES College of Pharmacy, Harapanalli, and PG in the year 1997 from same SES College of Pharmacy, Harapanalli, and completed PhD from Kuvempu University in the year 2006. From 2006 to 2007, he has worked as an assistant professor, and 2007 to 2022, he has worked as an associate professor, and from 2022 onwards till date, he's working as a head of the department of the JSS College of Pharmacy, Department of Chemistry of JSS College of Pharmacy, Mysuru. Coming to the academic achievements of the sir, sir has published around 140 international publications and 85 national publications, and he has guided 56 young pharma students and 3 are ongoing, and he has guided 10 PhD students and 6 are ongoing. Coming to the professional membership, sir is a member of APTI, IPA, and member of Royal Society of Chemistry, member of Society for Ethnopharmacology, and member of Board of Studies in Pharmaceutical Sciences, JSS, Aher, Mysuru. Sir has presented oral presentation at international conferences. That is in the 2011 at Malaysia, and Bangkok in the year 2018, and in the recent, that is in the 2024 at Dubai conference. He has presented 13 posters at various national and international conferences, and sir has worked as a convener for 5 workshops regarding hands-on training on analytical instruments. Sir has to his credit 3 patents and 6 grants. Coming to the awards, sir has received Best Oral Presentation Award in the APTI conference in the 2016 held at Manipal. Best Research Award from JSS College of Pharmacy, Mysore in the year 2024. Dr. P.D. Sethi Annual Award for Best Research Paper in the year 2013. Then CIPRA Innovative Pharma Research Award for poster in the year 2014, and he has received honor of appreciation from IJPR for selecting the papers for the APTI last year 2023 held at Mysore, and he has also honored for as an appreciation form by the JSS Aher for his contribution towards the development of the society in the year 2018. With this brief introduction, I welcome Gurupadaya sir. I request Honorary sir to present the bouquet and welcome the speaker. I request sir to take over the session.
Otherwise, I will
YouTube पे चलता है, YouTube पे।
ಬಟ್ ಸ್ಲೈಡ್ ಚೇಂಜ್ ಆಗಲ್ಲ, ಇದರಲ್ಲಿ ಮಾಡ್ಕೋಬೇಕು. ಇದು ದಿಸ್ ಇಸ್ ದ ಇದು ಪಾಯಿಂಟ್ ಅಲ್ಲ. ಓಕೆ ಸರಿ, ಸ್ಲೈಡ್ ಚೇಂಜ್ ಡಿಲೇ ಮಾಡ್ಕೋಬೇಕು ಸರಿ. So very good morning to all. So I think you are already participating from the past yesterday. I mean from the yesterday you are all attending for this FDP program. I think they are given a topic on the molecular docking and followed by the 3D printing. These are the two important topics it is given for you and all. Maybe in this particular crowd, I can able to understand that maybe the staff members are there, and maybe the faculty members, and followed by the PhD scholars would be there. I think so. That is going, because I want to know the crowd whether they are purely based on the faculty members and the interested chemistry team is there, so that my work is going to become easy. If suppose I hope that majority are chemistry staff only here. Chemistry staff. Okay, thank you. Thank you for confirming yourself. So regarding that, you are all belongs to a chemistry team, and maybe some other team would be there, because I know that some of whom faculty members are arrived from our own college, from JSS College of Pharmacy, Mysore, and they are participating for your well-known this FDP program, which is going to conduct for 3 days, 4 days. Maybe today is the second day, and maybe the 2 more days are waiting for you to spend on the followed by this docking is maybe today would be the last, and maybe the tomorrow onwards you are going to work on with 3D printing technology and all. Maybe it's a very good combination which is they made for you and all in making the showing you in the software based technique and followed by the 3D printing. Maybe currently both topics are both are interesting, whether you can take it consideration of the molecular docking and as well as the 3D printing. Maybe the Vgst has given the handful amount to the Arya madam, and congratulations madam for receiving a grant from the Vgst, and because of that I could able to stand here, and your participants are participated and they are came over here and to learn the theme based like your docking studies and as well as 3D printing. So moreover, I will be very much thankful to the principal of this college, Dr. Your A.H.M. Vishwanath Swami. Of course, he is my roommate and classmate, and of course, so we were studied in the same college, same college, that is in the SCS College of Pharmacy, Harapanalli only. Of course, so already they are given the introduction, so where I studied and all. So of course, so I was studied in the same college, SCS College of Pharmacy. So together we completed. Of course, sir has done your Vishwanath Swami sir has done in pharmacology, and I was done in M.Pharma medicinal chemistry. So we belongs to Gulbarga University only. M.Pharma our season first batch, and so we completed the M.Pharma together in the same college only. Later, I was worked as a faculty in the same college only, that is in SCS College of Pharmacy. For a period of 7 years, I worked there. So 7 years and plus 7 years of education, almost Vanavasa was completed, and 14 years in the same college. Later, I moved on to the Shimoga for a period of 3 years. I was spent for a period of 3 years in the Shimoga, where I completed my doctorate from the Coimbatore University, the Department of Biochemistry. Of course, Department of Biochemistry only, but work I did in the synthetic work only, where that was a very difficult situation. As you are currently doing and research on the medicinal chemistry or synthesis and all, it's nowadays becoming very trendy, and so it is a very easy going in selecting the molecules. So because of that reason, they are selecting the very renewable topic and very advantageous topic for you and all who are learning and who are going to start their career in the chemistry and all. But at the time while you are performing in synthetic molecules and all, and majority of our synthetic molecules are going to become dustbin only. The reason is that the synthetic molecules doesn't have any activities, and there is no use in taking into the next further level and making only the molecules, and majority of the cases they are all nitrogen based only. And as you know that when there is a nitrogen rich molecules are considered as nitrogen rich molecules are considered as a kind of fertilizers only, right? You are already aware that there is if suppose there is a molecules are rich with a nitrogen one, so then the people are saying, so your molecules may be better not for any pharmacological activity, it is better for the fertilizers only. So in that situation was there while we are synthesizing molecules at the time of in 1997 and all. I am telling the story of 1997. At the time whenever we are going for synthesis of any organic molecules and all, and at the time it was situation was different, but now the situation is totally different for you and all, because there is a even for taking the spectra of an IR molecules, we have supposed to move on to the CD-ROM facility where it is there in the Indian Institute of Science, Bangalore, and there we have taken the UV spectra, IR spectra, and followed by NMR and all. It was taken. So then that research was becoming so difficult at that moment. Now there is a situation is there in a such a way that in a fingerprint you could able to get a docking, and thereby you are going to reduce your work effort, and you can able to come out with a very good molecules, and you can proceed for your biological activity. This is how now there is a current trend is there, and maybe you are going to make use of this current trend, which is given an opportunity in the form of an Vgst, and maybe it is going to come in the ultimately we are going to expect any the research in it is going to publish in the renowned international or national publications only, where then only it is going to recognized, and of course it's happening. And while they are expressing that, so I was worked as an associate professor. Of course, I was not worked as an associate professor. And I could not get an opportunity because I completed after the completion of my doctorate. So I was there as an assistant professor in Shimoga. Then later, I was joined in 2007 to the JS College of Pharmacy as a professor only. So from the 2007, I was working as a professor in the same department. So in the last 2 years back, I'm taking the responsibility of the head of the department and all. And so I'm working currently, basically I'm the analytical person. But of course, my original degree is in M.Pharma, chemistry, PhD chemistry, synthetic molecules, bucket of the molecules. I was also synthesized, okay, chemistry man. But later, when I went into the Shimoga, where there is a scope was given for analytical one, and later when I was started my journey in the JSS, where I got again there is an availability of the advanced instruments, where maybe a kind of an LCMS instruments, and currently we have an LCMS instrument is there, NMR instrument is there. So what else you want to do in advanced research when there is a Schrodinger is there in your hand? I hope that this is an advantage which is there for current people, and making use of that advanced the facilities, and we can go ahead with making the effort in producing the good molecules, or maybe the good research, and all is possible. So with this the brief introduction, I will move on to the topic which is allotted for me. So that is accelerated molecular drug discovery, okay, and spotlight on docking and dynamic simulation. I think already maybe I saw that. So Madam Arya Madam is sharing the topic. What is the previous the speakers are delivered the topic, and what are their content was delivered, which I am observing it. So I come to know that already docking is already they made the utmost docking by the speakers. In the yesterday speakers, they made it. If suppose the docking is done very effectively and received, then it is good only. So because ultimately the docking is only the expectation from your end. So to dock in the KLE College of Pharmacy to understand the thing very well only. Okay. So with that, there is a so here in my presentation, I am planning to cover the introduction about this docking only again, then followed by the need of the drug discovery, and then followed by virtual screening and type of virtual screening, then molecular docking, and so again in that there is a classification is possible. Then maybe the molecular dynamic simulation was not covered, right, madam? Molecular dynamic simulation was not covered. Molecular dynamic simulations and challenges in the computational screening and advantages of this CAD, then followed by have some research scholars with me, and we are doing currently by utilizing this Schrodinger facility and all, and we are have some research work is there, so that I am going to explain in my presentation. This is how I am planning for the today's presentation. Okay, I think the topic which is given for is in docking, and again I am also going to take in docking here, maybe hoping that it's not going to become bore you, and maybe so I will not take the much time in giving the introduction about the docking and all, because you are already well aware about the docking. Maybe the yesterday's maybe some of the software was used to understand the effective docking, molecular docking is possible, and in that way I would able to give the brief introduction, and thereby I'm going to reach your the simulations and all. So that is what maybe you are all going to expect that. So later on and all, you are going to reach into the some other component where it's possible. Okay. And coming to the introduction about this drug discovery is concerned, as you are already aware that maybe this particular the things everybody is showing. So that is a drug discovery, how it is going to follow. So without this, the slide, there is no any drug discovery is not possible, because usually drug discovery is going to make a period of around 15 to 20 years is possible to any molecules to come out in the market. But fortunately, Indians are not producing any the significant molecules into market. Maybe you are aware that. So generally, we are going to depend on the molecule which are made the credential by the any the foreign agency only, and still we could not, even though we have so many strength of the populations in India, still we could not able to make our own new molecules, which has such credential is not happened. Okay, and now we'll make our level best. So in producing such an the wonderful molecules which can in further it can come into the market. Okay, and as you know that there is a drug discovery is going to take into its own time, maybe it may be in 10 to 15 years of time, and in which there is a the period in which where we can able to see the traditional drug discovery, that is especially the target identification, target validation, lead discovery, lead optimization, and clinical studies. So this is taking almost the 15 years of time, and this 15 years of time which can able to minimize by using the current trend what is taken for your study, that is a CAD only, that is nothing but computer drug design, the facilities which can able to minimize, and we can able to stop the physical synthesis of the molecules. So that is by using an either bioinformatics, or by using the target druggability, or maybe by using the talking studies, or by using the QSR model, and by taken into the ADM predictions. So these are all the virtual things are available nowadays. So by using this particular concept, you can minimize the duration which is required for the molecules to come out in the market. And there is the thing is available here in that usually when we are synthesis time and all, so where we are synthesizing the molecules, and physically we are synthesizing so many molecules, and that molecules may be reaching into the biological activity is very rare. The reason is that, so maybe the molecules of not target approach was there. Nowadays, everybody is approached with a target only, and because of that reason, again here in this case of your docking, where it is an involvement of target approach, nothing but we are going to target into a particular proteins or maybe particular enzymes. So thereby our requirement is going to reach us effectively, and the concept can be explained very well only. So in that reason, so where the importance is there for the drug discovery, so the reason is for, so we can able to see the important in the medical needs, where we can able to bring in wonderful molecules into the market, then improving the existing therapies where the molecules have some side effects which can be made into an effective way, and that is possible by using the drug discovery here, and the combating the emerging health threats. So that is another infectious disease and pandemics will be there, so which can be come over. So by taken into the help of this drug discovery, where there is a essential, and then followed by the important one will be our the development of personalized medicine. So this is a current trend is there for that, because there is a molecules are going to become waste, or sometimes we are could not able to reach the molecules into a particular target. The reason would be there is a improvement in the personalized medicine. So this is a current need for the any particular the molecules, are where we are going to synthesize, or where we are going to target for a particular proteins. So there is a requirement of the molecules for the personal medicine is there. So that I think this is coming the hub now for the booming for the current molecules, where we can target into a personal medicines only. And again, this is again maybe the important slide, or the slide maybe you're already the site again, which I'm showing as an introduction slides are for you also, where there is a docking is going to happen. For every docking and all, there may be a requirement of a very good software. Maybe some of you are maybe operated the software. Okay, and here in this case, what we are going to call it as a virtual screening only. Pharmacology screening, there are some pharmacologists would be there, so they are going to do the pharmacological screening, so thereby they are going to achieve about the molecules and molecules activity it's possible. But now there is the screening, we are going to do it virtually. So the virtual is going to become important word when there is a pandemic is started, where there is a virtual things are many things are happened nowadays. So of course at this level we are in the sitting physically, and we are talking about the virtual thing. So that I'm very happy that we are not making this particular FDP online. There are many things are going on in the online only. Still there is a FDP is fascinated, or I don't know. the things are going in online only. The people are still managed with the content and delivering with the habit of the virtual the mode the presentations are going on. So that is all depending upon the what is the type of the topic which is deliverable one. Maybe a kind of the topic which is taken for this molecular docking and all, maybe the virtual screening or maybe the physical based activities maybe gives the more effective the things here. The thing is that maybe there is the yesterday speaker or maybe the today Dr. Somajatta is there, and they are also going to bring in the effective way where you can able to reach us the requirement of the research through their software only. Okay, and here in this case, the important in the early stage of drug discovery, this molecular that is nothing but your virtual screening, and the purpose is to enhance the drug discovery process by reducing the number of compounds that need to be physically tested. So this is most important, that so majority of the cases we are going to synthesize the molecules and which is not going to reach its desirable activity. The reason is that so we are not taken into consideration of which is exactly the target is there or which is exactly the disease is there, and if we not taken the consideration of the important target or important protein or important the application, so our synthesis molecules are going to become waste only. Of course, so that was the era was there at the time, there is a things are going to become waste, but nowadays every single molecules you can able to bring it within virtually and proving that so this molecule is very better, and later we can go for synthesizing molecules. So that kind of a situation is there. So that kind of the opportunities are there for us, so that we can follow this one whenever we are going for synthesizing any molecules and based on by using this. So of course, so thereby we can able to minimize the synthesis, or ultimately we can minimize the chemical load on the earth only. So ultimately the many molecules, if it is not well taken into the consideration, it will be going to waste only, and thereby it is going to become chemically polluted. So that completely we can eradicate it by using this virtual screening only. And maybe now regarding its importance and advantages of your virtual screening, maybe you are already aware about this, the particular the concept in the case of your virtual screening and all. So through this we can able to understand the concept very well in terms of either you can consider this virtual screening advantages and importances. One would be in its efficiency or diversity or precision and safety, where in case of your efficiency, so we can able to reduce the time, which I already told that. So you need not to take your spend your time. So just you can have an important software where there is a online software is available, and later there is a paid version software is also available. That is all depending upon how your college or your management is supporting for your the software. You can able to utilize accordingly. Otherwise, there is things are available where you can able to directly visit into the particular center and you can carry out your training and all. So that's how they are bringing in the awareness. This particular the kind of FDP, especially those who are joining as an faculty members into a college, and they want to know, they want to learn, and they want to go for a higher level, and they want to achieve, because achievement is one of the important aspect whenever you join into any particular college and all. So there your higher authorities are expecting lot, and when you are having such kind of the target is there on your mind, and when you are having such kind of the influence is given by your higher authority, in such cases we have to be give the better result to that academic or to the college, and by utilizing this are all the facilities we can make use of that. Okay, and here in this case where there is an we can able to reduce the time and compared to the traditional methods of the screening large compounds, and where you can able to bring the diversity here, we can able to scan, that is, you can screen the vast libraries of the compounds, including those with diverse chemical structure. Of course, already maybe you are already aware about the different type of the software which is available in getting the molecules or the structure and all. Okay, and what are the different types of protein which are available and all. So by if you could have such kind of a wonderful library with is available, and we can certainly come out with using this docking studies, and we can come out with the even by minimizing the toxicity also by using the ADMA facility which is available in the docking, and we can come out with very good molecules here. Then coming to the precision, identifies the compounds with an higher likelihood of binding to the target and increasing the success rate in the drug discovery. ultimately everybody is working on the drug discovery only, because we want to have our own molecules, and that particular molecules has to be bring into a kind of a patent. So there is a question is going to arise here, whether I can go for publication or whether I can go for the patenting. So these are the very important issues whenever research scholar is entering into the preparation, or when he's having a handful molecules in their hand. Maybe this is an important the decision the researcher has to take it while he wanted to proceed for his research. There are two things are going to happen here. So either your research can be protected by making the patent, and the another thing is going to happen in the case of publications. So this is mainly whoever is doing the PhD, or those whoever is doing in research. So they are thinking that way, whether I can protect my molecules, whether I protect my research work, or whether I make in publication in wonderful journals with an impact factor of 1 and 2 and all, or with an impact factor of 5 and 10. So these are the two things are going to come in the mind whoever is doing the research after the fruitful come outs. This I am telling that when you are already started your work and you have a very handful work is there in your hand, and what to do next, whether you can make it to publications into a good journal, or whether I can protect it into a patent. So thing is that, so again, when you are going for a patenting, and you need to wait for some more time, because your molecule should not be disclosed unless otherwise after the realization of your patent is published, okay, or patent is granted, at least at that particular time you need to wait, then you can go for the publication. This is how we can able to, but that is taking its own time, making your submission of your getting the art of work, and getting the novelty of the work, then followed by the particular agency has to give the humble report that your molecules are novel known by comparing with the literature. Then we can able to conclude that your molecules or your research is a novel, which is given by the score. Based on that, you can able to go for the patenting, and this how there is a research is going to happen. So this is going to take its own time, but parallelly you will be in an hurdle that, so your management may be forcing to make the publications, because you need to have the protect your molecules and you need to do the publications. Maybe in that case, maybe my suggestion that you should have a diversified molecules. One, you keep it reserved for your molecules for a patenting purpose, and one set of molecules you reserve it for your publication, so that you will be come out with the good molecules with then protecting the molecules with an diversified pharmacological activity. Keep it for your the patenting, where